So that's, let's say, a timeline to producers to get to know, let's say, well, if you tell me when is the most appropriate time to help me to improve marbling deposition, when would be? So that would be from the last trimester of gestation up to 250 days postnatally of the calf. So that's a kind of timeline that's more effective to increase the number of those fat cells inside the muscle. That will be later on at the finishing phase being filled with lipid and increased marbling deposition.
Welcome to this episode of the Beef Podcast Show. My name is Dr. Stephanie Hansen. I'm a feedlot nutritionist at Iowa State University, and I'm excited to introduce our guest today, Dr. Marcio Duarte. He brings a wealth of knowledge and experience on skeletal muscle growth and metabolism, both pre- and postnatally, and its overall impact on the quality of meat. His academic achievements include a bachelor's degree in animal science from the Federal University of Viçosa in Brazil, and a master's and PhD focused on meat science and muscle biology of ruminants. He joined the Department of Animal Biosciences at University of Guelph in 2021 as an assistant professor in meat science and muscle biology. So welcome to the show, Dr. Duarte.
Well, thank you so much for having me here today. It's a pleasure to have that conversation and extend some knowledge today.
Excellent. And I'm really excited about some of the things we're going to talk about today. I love bridging things like nutrition and muscle development. And I think that's one of the things that we're going to get into in this conversation. But before we get started, why don't you give our listeners a little bit of your origin story? How did you get to this point in your career? How did you develop a passion for the beef industry?
Right. So I came from a farm background back in Brazil. Actually, my dad has a dairy farm, very small dairy farm, and I got into livestock production because of that. I came from a small town in Brazil that it's a very strong university in agriculture and one of the best animal science programs in Brazil. And that's I started my career, my passion about animal science since my undergrad. So I got into school and I graduated in 2008. And before I finished my BS, I spent a year at University of Minnesota as an internship there as an extension program in agriculture, and it just boosted up my passion for agriculture. And that's when I got a little bit more about what I'm gonna love and what I'm gonna do for a career. And then I came back to Brazil, and then I had the pleasure to be advised by Dr. Pedro Veiga, who is a big name in the industry right now, and he works at Cargill. He just moved from the academia to the industry, and then worked with him during my master's and my PhD. It just got me amazed about how the beef industry are big in South America, and now I just came up. I started as a new faculty back in 2014 at University of Viçosa. I finished my PhD and 1 year of postdoc in animal biotechnology lab. And then I stayed there working with beef as a model to understand a little bit more about growth and development of the muscle and also meat quality. And after 7 years back in Brazil, I just decided to try something new and a new adventure. And then I end up being here in Guelph today. What attracted me most in North America is how we can get a lot of information that sometimes is a different system. It's different production system actually here in North America compared to South America. And that's what actually got me more interest to come and try to do some research down here as well, just because of the new challenges and new things that I would seek as a researcher using different systems. So that's a kind of short, long story short, but that's how I end up being here in Guelph and doing research using beef cattle as a model today.
Awesome. Am I correct in remembering, did you have a period of time where you were at Washington State?
Perfect. Yeah. So during my career, again, I spent like a month also during my master's at USDA Mark in Nebraska, because at that time during my master's, I was just starting off understand about meat science. And I started reading a lot of papers for protocols to assess meat quality. And then I contact Dr. Tommy Wheeler, and he was really helpful. We exchanged some information at that time. And then after like 15, 20 mails I sent to him as a math student, he goes like, well, why don't you come over here and then we can teach you all the techniques you need to know. That would be a lot easier for you as well. And at that time, my PI, he was starting a new program in mid science in our department back in Brazil as well. So I thought that would be a perfect fit to spend some time there and bring all the techniques that we need in the lab. And then I finished my master's, and I got a lot of questions when I studied mid science by itself. Like, okay, we got that as a result, but what actually makes those changes? Then that's what it brought me more towards to the cell biology and molecular biology. That like, well, that's what kind of does a lot of the job here in terms of changing the traits we see in the meat. Then I decided to go over to muscle biology and go using a lot of molecular biology techniques to understand those changes. Then I started my PhD program, and by that time I had an opportunity to go to Denver for ASAS meeting, and I watched a presentation from Dr. Mindu, and it was back in 2010, and it was a presentation that ended up being a review paper published in Journal of Animal Science. That's one of the highest cited paper, I guess. And then I got amazed by that. So that explains a lot of doubts that I had that time. Then I contacted him like, well, we do have an opportunity to do a research project back here. Are you interested to have me as a student in your lab so I can do part of my research in your lab? And he was like, yeah, why not? Then I ended up being in his lab for a year at Washington State from 2012 to 2011, 2012, before I came back to Brazil and finished my degree there. So that was kind of the whole path of my master's and PhD.
So I know Dr. Du obviously has a strong reputation as somebody who's very interested in the molecular drivers of marbling. And I'm guessing that that has been one of the things that really shaped your research interest there in your lab now.
Mm-hmm. Yes. So we had one really good chance of being at Washington State to use Wagyu as a model for our understanding of marbling development. So while I was there, we came up with a research project that was part of my PhD actually, and it was one of great chance to work with Wagyu, which is not very common to have a Wagyu herd in universities to do research. So and then after working on his lab, and I got a really good passion on cell biology and try to understand what actually shapes the marbling development. And I have been using this as one of the main cores of my research right now. Yeah, for sure. I've been doing this for about 9 years now since I finished my PhD.
Excellent. Okay, so we're going to talk a little bit about some of the specifics that you are working on in your research program right now. But I think, correct me if I'm wrong, but I think it might be helpful if you could give our listeners a little bit of a 101 style lecture on some of the changes that are happening to the fetus during the phases of gestation. So thinking about like pre-adipocytes and things like that, right? And that are going to later have impact on that calf's ability to grow, his ability to marble, carcass quality, because I think that's some of the things you're going to tell us about. So maybe can you tell us a little bit about what are some of the, the timeline of those changes in development and things like that happen during fetal development?
Right. So one of the things that we usually look at the pregnant cow, and trying to do some interventions. And of course, nutrition is one of the most common interventions that we can do, is because during the fetal development, during the pregnancy, there is a lot of going on in terms of commitment of cells that undergo either to form the muscle or to form other tissues. So, and within the muscle, we do have the muscle cells there, but we do have a lot of other cells too. And one of the cells within the muscle that are developing there and trying to get their job, let's put it this way, during this time, it's what we call the adipocytes or pre-adipocytes, which actually are fat cells in a very early stage of life that will be later on filled with lipid and help to develop the marbling. So because when you look at the animal during from gestation to slaughter, it's not too long. If we try to work on those animals postnatally only, mainly at the finishing phase, sometimes it's not time enough for the animal to, boost up and put more intramuscular fat or marbling deposition. That's why we end up being like, what happened during the gestation time, and what is the time frame, the best time frame that we can work on at this stage to improve the number of these cells at that stage. Once the animal are born with more cells, it's a huge probability that these animals will have more marbling at the end at slaughter. So during the pregnancy, we usually work on 3 trimesters stages, right? And one of the main stages that we're looking at right now is the last trimester, the late gestation, because that's when most of the other cells and also the muscle cells are already formed. So they're not, the adipocytes are also being formed there. But in terms of priority, the adipocytes would have less priority than other type of cells developing that time. So if you take care more about the cow at this point, and do some manipulations on the dietary changes, or trying to use some compounds in the diet, we may be able to trigger some signals on the fetuses that will help those cells to undergrow to form more adipocytes than other type of cells. So let's say, to summarize it, we do have the 3 trimesters, and the very first one and the middle one is more towards to muscle development, muscle cells development. And the last trimester, it's when we have the timeline that a lot of adipocytes will be start to be formed inside the muscle, and that those cells will help to increase the marbling deposition and the offspring.
Awesome. I think that's really useful for producers to think about that. Basically, the first 2/3 of gestation being where we maybe have the most impact, where we need proper nutrition so that a cell deciding to become a muscle cell versus a neural cell isn't saying, well, I don't have any nutrition. I better be a brain cell over a muscle cell. Like I've got limited choices right now. So you want to make sure they can make that decision versus that last third of gestation where they're starting to basically lay the future groundwork, right? If I'm a pre-adipocyte at this point, then later I can be a lipid-filled cell, and that's our good marbling. And this extends beyond calving, right? So we also have, what is it, up to 200 and some days after that calf is born that we can influence some of that adipocyte development. Is that true?
Yeah, is that correct? So the concept of what we call now marbling window, back in 2013, I had the pleasure to participate in one of the review papers that it's published as well at the Journal of Animal Science, that it's a concept that shows, based on the literature and a lot of research that was done by a lot of researchers here in North America and around the world, that have shown that not only during gestation period, but also at a very early stage of postnatal period, we are able to increase the number of pre-adipocytes as well. So basically, there are one population of cells that when the animals are born, they're still uncommitted in the muscle. So those cells has no job at that point, and depending on the signals you give to them, they can get a job as a adipocyte. So they can become adipocytes. So there is one process in biology that we call hyperplasia. So in this case, we're increasing number of cells. So after birth, up to 250 days, we're still able to give those uncommitted cells adipocyte jobs. So by feeding the young calf properly, we're still able to get those cells committed to become pre-adipocyte. So that's, let's say, a timeline to producers to get to know. Let's say, well, if you tell me when is the most appropriate time to help me to improve marbling deposition, when would be? So that would be from the last trimester of gestation up to 250 days postnatally of the calf. So that's a kind of timeline that's more effective to increase the number of those fat cells inside the muscle that will be later on at the finishing phase being filled with lipid and increased marbling deposition.
So when you were talking about these cells like they didn't have jobs yet, I just pictured like our undergrad students coming to college. They don't have jobs yet, but they're going to have all the potential in the world to go into a job in animal science or a job in ag business or whatever their training ends up being for them, right? So I know that this is an area you've done research on. What are some of either the nutritional strategies or other kind of strategies that you have worked on to say, okay, I've got a calf on the ground, or I'm thinking this last third of gestation. I know you've studied both of those things that can help move some of those calf cells towards making that commitment to decide to be an adipocyte.
Mm-hmm. So a lot of research that we have done before in the past have shown that the potential of increasing different nutrients at the dam during the last trimester that can help the calf to improve more adipocytes, intramuscular adipocytes. So last year we just finished a trial and have published one research paper showing that one of the main things that we have seen so far that it's more effective, it's when you work on protein and amino acid diet side of the diet, let's put this way, that can actually, it seems to be really effective to give some signals to those uncommitted cells to undergo to adipogenesis. Last year, again, one of the papers that we have published, we learned from a trial that we did. We tried to use an we increased the levels of protein in the diet that's kind of protected from the rumen and goes towards to high passage levels of amino acids towards to after the post rumen, trying to use that as a vehicle to deliver somehow more amino acids to the fetuses, aiming to see whether those amino acids are maybe act as a signal for those cells to become what we need. And what I learned from that trial is that by feeding high rumen protected protein in the diet of those cows at late gestation, we were able to see a quite nice increase in the commitment of those cells to adipogenesis, which means that those calves were born with more pre-adipocytes and actually the control group that received the same not enhancement on rumen protected protein. So protein has been one of the things that we're looking at, but more specifically now, there's a lot of research going on looking at specific molecules or biocompounds that can be used in the diet as well. So after birth, one trial that I participated in collaboration with the researchers in Brazil, we have seen that by using vitamin A very early stage of life, when the calf was born and it was injected with vitamin A, we were able to see a nice improvement in marbling deposition at slaughter of those calves too. So, and I know that vitamin A, it's kind of tricky thing because sometimes at the early stage of life, it does one thing that can help to improve marbling. And when you use vitamin A, high levels of vitamin A at the feedlot, you can actually do the opposite. But again, that's another thing that we have seen and learned from research that can help increase pre-adipocytes at that very early stage of life. So I would say that right now, specific biocompounds, there's a lot of going on in the industry right now, isolated amino acids and rumen-protected amino acids that can be very, very useful to act as a signal for those cells to become what actually we expect it to become. So I would say that in the next 5 years, we will be more precise on that side and actually just make global changes on the maternal diet, trying to see what happened.
So let me ask you a few follow-up questions on when you're thinking about having maternal diets that have more bypass protein. Was that do you think it's a specific amino acid that is a part of that, like that composition of whatever you were feeding? Or do you think it's more just about, excess energy availability as they're catabolizing those carbon skeletons? Is it a nitrogen signal? What do you think the mechanisms are there?
So from the trial that we did, what we observed that by feeding more the bypass protein in the source we used at that time was soybean, we saw that we increased a lot of the arginine levels of circulating levels of arginine in the cow. And arginine is something that can really boost a lot of things, right? It acts as a signal amino acid, and also for the cow, it helps to increase the vascularity of the placenta that will actually help to deliver more nutrients to the fetuses. So it can be both. So we were not able to discern what caused specifically the change that we saw there. If it because we increase the vascularity and more blood flow to the fetus and deliver more nutrients to the fetuses, or if it was because the arginine itself was causing those signals changes at the cellular level of the fetuses that got those cells changed their phenotype. But I would say from what we learned so far, by feeding them, it looks to me that one of the main amino acids that I think we need to take care of now, that would be arginine, that can help for sure improve those both sides. And that's what we have been doing as a following up research after that now, trying to increase the arginine levels somehow in the cow and see whether there are effects on improving marbling deposition at late gestation.
Okay. Well, we know there's some rumen protected arginine products available, so that would be, opportunities. I'm intrigued because arginine, like you said, is kind of a complicated little molecule. It affects a lot of things. So it also has kind of a delicate balance with like nitrous oxide production, right? And so which is actually like a gaseous transmitter in the cell that can be, that's probably where some of that vascularity stimulus comes from, right? Like, so increasing that blood flow to the placenta.
Yes, we tried once to work with rumen protected arginine, but we were not really successful in getting some source that can guarantee to us the levels that would be protected or not. I think at the industry right now they're working on the best method to protect that specific amino acid. That's what I talked to some of the folks 2 weeks ago in the conference. But what we have been doing so far, trying to increase levels of arginine, is trying to use another molecule that can boost the biosynthesis of arginine, or use some other compounds that can actually act as an arginine sparing molecule. So inside of the cow, we're not feeding arginine directly, but we're feeding something that can actually increase the levels of arginine. And I think, like you said, I think it's correct to say that by increasing the levels of arginine, we we do have some things going on, like nitric oxide increasing, that can actually act as an angiogenesis boost the blood flow overall through the placenta to the fetuses. And but also again, it can act a specific molecule acting as a signal at the fetus's level. So that's something for the research wise. Using the livestock is quite tricky to do, because sometimes we're not having much access to the fetuses itself, unless we use other lab models sometimes that not picture what we're looking at. But I think that's one question that the next few years we're looking at, to see if increased arginine levels, are we just improving the nutrient delivered to the fetuses, or are we bringing some other signals specifically to those cells at the fetal stage that can help to increase marbling deposition?
So, is this some of your work with the guanidine acetate that you've been working on to work on some of the arginine sparing? So, you want to tell us a little bit more about that?
Yep. So we just finished a trial a few months ago, and I was fortunate to get a presentation at the conference, the American Association conference, 2 weeks ago in Albuquerque. There, we have a great experience sharing some knowledge with some folks that are interested on the topic too. And we fed those cows. We got a trial that one of the groups we fed some high levels of guanidine acetate. And guanidine acetate, it's a molecule that can be produce itself in the organism. And for those that are on the audience right now that like I do not know much about guanidine acetate at all, but just to summarize it, it's a molecule that it's produced by the animal organism itself and actually helps to the muscle to increase some source of energy. Let's put this simple way. But it also causes some other changes too, because sometimes when you feed that molecule that was isolated and put in the diet, we not only increase that energy production in the muscle, but we also cause some of the metabolism changes that can save some molecules like arginine. So by feeding guanidine acetate in the diet, the organism of the animal will not utilize arginine to become guanidine acetate. So we can save some arginine. And then this arginine itself can do the job that we're looking at. So that's one of the research that we're doing right now. We've in that experiment we fed those cows guanidine acetate, and we had a control group that guanidine acetate was not added in the diet. And the results we got so far showed us that we were able to spare arginine quite a lot in those cows. And whether these arginine sparing effects will increase or do some changes at the fetus or and the calf, we're still ongoing that part of the research. But the results we got so far, we were able to see that actually it acts as we expected. And I mean, it was not a new thing. It's been done on other livestock species, but it's kind of useful thing to use that approach in pregnant cows. And that's what we did at the last trimester of gestation, because we're trying to manipulate in the same timeline that we can try to improve the intramuscular fat deposition. So that's what we have been doing so far. We do start another trial like a month ago, looking at guanidine acetate levels, different levels now, and combined with some other rumen protected amino acids to see whether we're able to change the cow metabolism towards to what we actually want to.
Yeah, I'm kind of reminded of some of the work that like Alison Meyer did up at NDSU when she was working on fetal programming using a lot of sheep models. And they were looking at things like selenium and then energy restriction and protein restriction and things like that. And if I recall, some of her work came back to showing positive effects of, I want to say arginine was involved in it, but it was like showing like placental blood flow differences and stuff. So it'd be interesting to think about whether like testing your hypothesis in like a sheep model or something where you could get twins born and you could be able to look at one at birth and do really intensive kind of molecular studies and then have the other one to grow out and be able to look at things. I mean, I know marbling would be very different and stuff in the sheep versus calves, but just kind of interesting to see how some of the fetal programming work, quite a bit of it is done, kind of has been done in sheep.
Right. Yeah. Well, definitely. A North Dakota group, and I have read a lot of papers. I follow them since I was a PhD student, and the work they have done, they're kind of amazing. And we learn a lot from what they do as a research, and we use them, their findings, to try to, evolve in our research here too. And I think that there, I, if I recall correctly, I think they have done some work on arginine, rumen protecting arginine on sheep model. And the only problem, we tried to use that in the past, but again, some of the companies in the industry, they have the power to, come up with some protection strategy and all the technology for that. It was quite hard for us to find, a product. That's why I think there is no arginine, rumen protecting arginine in the market yet. So they're trying to see whether there are the best method to rumen protect that. So that's why we have not end up being using rumen protect arginine. But some of the idea that we got, for sure, it was based on their findings. So I think we need to explore a little bit about what actually happens at the molecular level. And the main idea of my research lab is trying to understand those molecular mechanisms underlying these changes. So once that's our focus now. Once we understand what's going on with the commitment of those cells, we can come back and say, okay, so we fit this, and that's what actually will cause those changes. Just like for human now, for example, everyone know after a lot of research back in many years ago what folic acid does, and that's why the pregnant woman needs to, supplement herself with some folic acid based on what not only the changes but the mechanism behind it. So I think we're reaching that stage very soon. Not only my lab, but I know that it's a lot of other labs looking at the same thing right now. And I would say that in the next few years, we'll be more precise in terms of what specific biocompounds that we can use to uh, to do the job that those cells we want to get them to be doing.
So, we talked about the fact that there was this window that extends beyond once the calf is born out into 250 days or so of life, where we could still influence the potential for future marbling. So, my next question is if we move past the kind of fetal or the gestational treatments and we've got a calf on the ground now, are there studies that you've done looking at that early calfhood potential intervention with nutrition to set them better up for success for marbling?
Yeah, we did one trial trying to use one common approach that's mostly used for all the cow calf operations, using creep feeding of calves to see what what actually cause at the cellular level in terms of increasing or not marble intramuscular fat cells commitment. And why we use that approach, we're not looking at specifically one diet or specific one nutrient, but we just use that common approach to see, because it's a common sense now that when you use creep feeding to the animals, we get better results, we get better carcasses at some point. It really depends on what we do after the cow calf season anyway. But for those that are do the whole thing, like from pregnant cow to get the calf on the ground and slaughter those calves later on, I think it's a useful tool. So and then we use one group of cows that was crab fed, and the other group was not crab fed, and we collect the tissues from them to see, for those calves, to see what actually happens in terms of signal and molecular changes at the skeletal muscle tissue, to see by applying this marbling window concept, are we able to actually increase the number of pre-adipocytes. And what we learned from that trial is that, first of all, we did not increase those unemployed employed cells, like that has no job. That's one of the hypotheses. So those calves receiving more nutrients, they would increase the population of uncommitted cells, those cells that has no job yet. And by increasing them, if they have the same ratio to go to adipogenesis, we would be increasing intramuscular fat deposition. But it was not the case. And then we questioned ourselves, like, okay, if you're not increasing that population, are those nutrients being delivered, triggering somehow these cells to undergo more to adipogenesis or not? And that's what actually we saw a change there. By supplementing more nutrients to the calves, from 100 days of age to 200 days of age, we were able to increase pre-adipocytes, intramuscular pre-adipocytes, and one of the and then we investigate a lot of molecular mechanisms behind it to see, okay, so why those cells now are going towards to more to pre-adipocytes. And it was a lot of molecular work done on that trial. We just published 2 weeks ago that paper in Meat Science Journal. But what we learned, what I think the bottom line for the producers to hear that is that we were able to use that crib feeding system, of course, to increase better carcass, to increase carcass weight, to improve the carcass quality at the end. But everything starts from the beginning. So it was kind of proof of concept trial that we did, and actually we saw that we were able to increase the intramuscular pre-adipocytes by feeding them, providing them more nutrients from 100 days to 200 days of age, 250 days of age.
It seems like the commonality between both your gestational work and your kind of creep feeding work is excess nutrients and or getting nutrients to that calf at a critical time.
Yeah. What we're trying to understand now is that, okay, so we're feeding them and providing them more nutrients. And the next step is like, there is any, from those nutrients, there is one specific compound that you can use. So that will be more precise than actually just overfeeding them. And I think that's what research it's going towards now, to see if you can use specific molecules like even amino acids, for example, not only feeding them more, but feeding them what. I think that's the next question now that we need to take care of by understanding how we maybe manipulate those cells, and what actually the signals they're showing us they are receiving by providing them a lot of nutrients. We're able to come back. Okay, so let's try get what they actually need only to get what we want them to do.
Also, ultimately, our cattle have a lot of different depots of fat, right? So we've got the intramuscular fat, which is a positive thing and we get paid for it. We've got subcutaneous fat, which we get a higher yield grade and we get a dock for that from the packer, but we still have to have a minimum. And then we have visceral fat, which actually can also be a negative and can actually be very metabolically active and cause inflammation and other things like that, right? So do we know anything about signals that say we're going to make more intramuscular fat cells, but not necessarily set up the animal to just have more back fat?
Yeah, well, that's $1 million questions, I would say, right? Because yeah, definitely that's something for the research back in the past. I would say that at least for me, for the back 10 years in research, that I have been reading and tried to come up with some hypothesis about it, I've seen a lot of people with saying this, asking the same question: How can we improve marbling without increasing the overall fat of the carcasses? And one of the things that we're looking right now, we just finished a trial last week here in Guelph, and we got those calves slaughtered last week. That we're trying to use vitamin A again because of the potential of it. And I think we know too little about the potential of it. Uh, we expanded a little bit going back. We did a trial in the neonatal calf that we inject them, like I mentioned before, but we tried to push a little bit before that. So we fed high vitamin A diets. We had pure vitamin A on the diets of the cows, trying to increase and enhance the commitment of those undifferentiated cells to adipogenesis. And that is based on the research done right now. It seems that vitamin A, for some reason, it's more active on those intramuscular adipocytes and actually the other adipocytes in different fat depots on the carcasses. We are still ongoing on collecting the data here, but it seems that the intramuscular adipogenesis was slightly changed on the calves that came from the dams that received high vitamin A diets. It was not a big change though, but the overall thing and the main side of the project is, okay, when you get those calves slaughtered, are we able to see any quality grade increasing and not increasing the overall fats of the carcass? So that's what my student right now, she's working on the data because we just finished this slaughter last week, but we were able to measure different fat depots on the carcasses to see whether marbling or intramuscular fat was increased without increased the other carcass fat depot. But again, there is a lot of research that I was involved with when I was at Washington State working with Dr. Mindu and also Dr. Mike Dodson. That we were able to see that for some reason intramuscular fat adipocytes, they respond differently than adipocytes from other fat depots in the carcasses. So right now I would say that because of, again, all the results in the literature right now about vitamin A, we speculated that vitamin A would actually differently on those adipocytes than other adipocytes. And that's another ongoing trial that we're going to do now in vitro. to see whether we are able to understand those mechanisms and how those different adipocytes respond to different signals in the carcasses. Again, that's a lot of uh, every time we finish a research about that topic, we came up with more answers, more questions than answers, unfortunately. But that's how research goes, I guess, right?
Absolutely. I think that's always true. I wonder on if some of like the cellular machinery is different between intramuscular fat cells, subcutaneous fat cells, et cetera, because like some of the work that like Steve Smith has done down at Texas A&M, where they've examined like how the different beta agonists affect those different fat cells. They don't like the intramuscular seem to be less at risk of being affected by beta agonists than some of the subcutaneous fat cells, which is, a positive. So we're not decreasing marbling when we use those products. But it just makes me think that there's got to be a profile that's different between those different types of fat cells, right? That's somehow maybe it's affected by the neighborhood cells, right? It's like, oh, I've got these extracellular matrix cells around me, and I've got, these complex muscle fibers, and it's harder to get there. There's less or more blood flow depending on the depot that you're looking at. Like, and then vitamin A itself is just kind of a tricksy little thing, right? Because it's got lots of different forms in the body and it affects everything from gene expression to inflammatory response and immune functions. So, yeah, but it's very cool to hear the potential positive effects of vitamin A on gestational impacts on calf marbling, because we know that late gestation vitamin A is the most likely time point to have a problem with vitamin A. We've either burned through the summer status boost by that point, or if it's like here and it's been brown half of the summer, my cows aren't getting much of a vitamin A boost this year. I know I'm going to have to start supplementing it sooner during the winter. So I think that's cool for producers to be thinking more holistically about the benefits of that, especially if they retain ownership and keep those calves.
Yeah, that's for sure. And again, one of the I always ask myself, there's one group of Dr. Bradley Johnson in Texas Tech that we learn a lot from the studies that he lead on the cellular and the cell culture side of things. Because one of the things that I think it's more tricky to understand how we may be able to manipulate those cells or not, it's when you work with the live animal. Because there's a lot of interference that we make a lot of assumptions at that point. Being honest, when you work with the livestock, that because marbling, it's a trait that it's not controlled by one thing or two things, but it's like a lot of things controlling it. And like you mentioned before, uh, even in the tissue itself, there's a lot of going on there. We assumed that it might be a straight effect on the adipocyte and not to the other cells, and get some side effects on the adipocytes. So again, Dr. Bradley Johnson, he has some astonishing work on using in vitro studies, in vitro models, that we kind of learn a lot from what he has been publishing so far. And I think one of the things that we're missing at the moment is to do some in vitro studies that we can actually get more control on those cells. And the tricky thing is that to work with it, to learn about the mechanisms of it, we need to use the cells that come from the model that we are interested on, which is not an easy thing to get. To isolate those cells from the livestock and get the proper cell culture ongoing, it's kind of a lot of work. It's not something that we're doing in mouse and rats. That's sometimes it's more easy and more fast to do. But I think that would be an additional trial that we're planning to do soon, is trying to use primary cell cultures to understand some of those mechanisms specific on the cells that we're looking at right now. And by combining both in vitro studies and using the livestock as a model, I think we may reach more knowledge in a short period of time that can get us towards the more precise nutrients that we need to add either late gestation or neonatal stage that can help us to boost marbling deposition.
I think this is such a great example of why, like in my research in feedlot nutrition and work like yours and others, it's so important to have these mechanism of action type experiments so that we can figure out why something happens, and then we can turn around and we can exploit it in future work, right? So instead of having to waste money on overfeeding to an extra condition score or something to get these positive effects, we can instead be very precise about where we spend our money. We're going to supplement this nutrient X that has this positive effect, and you can decide how much of it, and you can decide on the timing of it. So I think that's so cool to see how your work is an example of this, kind of going from molecular approaches to whole animal applications.
Yeah, and one interesting thing that you just mentioned just reminds me of something, because we always get asked by producers, for example, okay, based on the work you do, what do I feed my cows at late gestation? And then when you show the work and you try to, tell them what you did, we increase the global nutrients in the diet. So it kind of overfed them, or, is not a nutrient, one of the things. And then I totally understand that sometimes it's hard for them, like, okay, should I overfeed the cows? And what is the benefit of it? Uh, and that's something that we need to improve. And I think that what research will tell us in the next few years, not only provide a lot of nutrients at a time, because we already learned that we cannot restrict a cow at any moment of gestation. We cannot restrict a calf at young stage of life because of obvious reasons. So what we're doing now, we use that to understand what actually occurs at the cellular level, and by understanding what occurs at the cellular level, we understand what actually those cells need. So you go from the macro to the micro thing, and we're not doing trials restricting or overfeeding. Try to use that. Okay, you need to overfeed them. No, we are overfeed them to see what happens at the tissue, and by getting those answers, we may be able to, okay, now by not overfeeding, but if you feed one nutrient or a specific nutrients, we're able to manipulate what we want. So it's I always tell them when they get those questions, we do not have the exact answer you need right now, and unfortunately, but it's coming up soon, I guess. Again, if you go back in 2008, 2010, for those studies from all the labs that work with feeder programming, for example, and by now, 13 years later, they have a lot of more focus and nutrients they're interested on, and they have shown and proved that actually does the job and not the global thing anymore. So that's, I would say, again, in the next few years, we're more able to be more precise on that side. But by combining both, cellular and molecular biology tools, we're able to understand what actually happened in the tissue. That's something we're trying to focus on to give those answers back.
We have a time and labor saving product for you. Beef and Dairy Agrislat by Healthy Farms is your solution. No more lugging jugs around the barn every month. With Beef and Dairy Agrislat, you simply drop the slat through the floor twice a year, and it works to break down solids, reduces crusting and forming. To learn more, visit myhealthyfarms.com.
Okay, well, I think we've reached that time in our interview where it's time for our famous 3 questions. So are you ready for these?
I guess. I hope.
Okay, so question number 1 is, what is your favorite beef resource?
Well, my favorite beef resource is, honestly, I'm really science reading based guy, but the scientific journals are the main source for me. But for the beef side, I've been reading some beef magazines to get more towards into the industry here in Canada, since I came out from a different system. So beef magazines and agriculture magazines are the main source of the knowledge for myself, what's going on in the beef industry, honestly.
Yeah, I imagine that the beef production system is slightly different coming from Brazil, moving to Canada.
Oh yeah, I would say the main different thing is that the variability. So here in North America, it's more standard production. So when you go to the slaughterhouse, we see more standard carcasses coming in with some slight changes. When you go back there in JBS, big plants in Brazil, you see nice carcass, just like we see from Angus here in North America, to the worst carcass that comes from a dairy farm that is slaughter a cow that is not working properly for them anymore. So the variability there is huge, and it's a different system. It's a huge country, but it's doing an amazing job in the beef industry, I can say. So by facing more challenge, it teach you a lot of efficiency. So yeah, it's different. It's not better and then worse. It's different.
Absolutely. Every place has their challenges and opportunities.
Yep.
Okay, question number 2. What is something not related to beef that you are reading or watching or binging right now?
Okay, so I've been watching a lot of one of the TV shows that I like a lot right now, watching with my kids. It's something to teach them about Canada. It's N with an E TV show, and my daughter, she's 10 years old, and she got amazed by that TV show, and that's what I've been watching with her because it was filmed here, and she's learning the culture by watching that TV show. So that's what I've been watching at the moment.
That's awesome. How old are your kids?
10, and I have a daughter at 10, and I have a son 4 years old.
All right. So the third and final question is, what is a trait of someone you know that you think has helped them be successful?
I think it's persistence. It's kind of cliche to say that, but to succeed in something that you want, you need to have a goal, and you need to persist on all the steps to get into what you want. And that's something that I have been seeing in some of my inspirational people that I have met throughout my career. And I cannot say names here because there are a lot of them, but that's one of the things that I have for those people that have inspired me so far. That's what I saw, persistence, and that's what got them in a succeed career.
I'm just realizing now, but this is actually a really cool kind of way to mirror the beginning and the end of every episode, because we start with your origin story and you kind of drop some of those names, right? Of who were your mentors, people that you worked with and looked up to. And then here at the end, often we hear from somebody something about one of those mentors or something. So that's kind of a cool way to wrap up our time together here to be appreciative of those cool mentors that we've had.
Sounds great. Well, I appreciate that. And it was a great opportunity to talk a little bit about what we have been doing as research, and also, to reach out to people that sometimes it's hard for researchers and professors to get that language properly, because what we have been doing here is not a digestible thing sometimes when it comes down to producers. But I hope that helps somehow for those that are watching this episode, and get some answers or some more new questions that can come back to us at some point. And then we can explore as a research opportunity as well.
Well, great. Thanks again so much, Dr. Duarte, for being with us today.
Well, my pleasure. Thank you.